Hi all. Thank you very much. Thank you to Michelle and the team for the invitation to speak. Great to be here. I'd like to talk to you about a soft X-ray microscope we've developed in Dublin for imaging whole, fully hydrated cryo preserved cells. So soft X-ray tomography has been around for a couple of decades now, but it's been really the preserve of synchrotron facilities. And to date, there hasn't one can actually have in their lab. And therefore, the workflows potential workflows that can be developed that combine it with vEM or volume EM have not been available to date. So really, this is what we're really seeking to do with the availability of the lab-based system. We're aiming to propel it into labs worldwide, and that really provides scope to develop these novel workflows because the technique itself is inherently very compatible with EM workflows in the sense that it can provide 3D volume data context, I guess, to EM data sets. So I'll aim to show you a few examples here. Hopefully you'll get the idea. So this is the obligatory opening slide. So when we talk about soft X-rays, we mean low energy X-rays. So they have an energy between about 300 and 500 electron volts. That's between the oxygen and carbon absorption edges. So within this energy range, basically the X-rays are relatively easily transmitted through water, but are preferentially absorbed by the carbon-containing organelles. So essentially, if you illuminate a fully hydrated biological specimen with these low energy X-rays, you can produce a map of the organelles right out of the box. So essentially, the organelles appear dark against the aqueous background. So we call this a really unbiased, it's a truly unbiased view of the cell. We can image up to 10 microns thick of specimen to a resolution of about 55 nanometers with our device. So yeah, fully hydrated whole cells, no staining, no chemical fixation. So it's a totally label-free technique. It can be combined with fluorescent imaging, as I'll show, and of course, electron microscopy as well. So we call it a non-destructive 3D imaging technique. So just to recap, synchrotron beam lines such as the B24 at Diamond across the road have had this technique available for the last couple of decades, and 4 or 5 other synchrotrons worldwide. So our system is on the right-hand side. We've developed a compact light source, which is really the key innovation, the innovative step in terms of bringing this from the synchrotrons to the lab. So the footprint is about 3 by 2 meters. I'm not going to go into the technicalities of the microscope here. I'm really focusing on the workflows that we're developing. So we have a system functioning in Dublin in our labs at the Sirius XT lab. And our first off-site deployment happened last August. We have a system at University College Dublin in the Biomolecular Imaging Institute there. And we're also operating the microscope, and we have about 40 research groups at the moment currently using the microscope in some shape or form. And we're developing correlative workflows to combine it with, as I said, fluorescence, electron microscopy. Our system is compatible with a range of sample holders. We've recently had the Joule FibSim holder in the microscope. So yeah, so we're really exploring these various variations, I guess, on a theme in terms of inserting soft X-ray into CLEM and volume EM. So just as a quick overview slide, I mean, we can image through, as I said, up to 10 microns of fully hydrated biological material. We have a field of view of about 30 microns. That's 30 by 30 microns at highest magnification. So we can flip through the slices and we can pick out organelles such as mitochondria, lipids, endosomes, lysosomes, etc. You can pick out virtual slices from the stack. And these are some of the organelles we can readily pick out. We also have an integrated fluorescent microscope. It's a basic wide field system. It's in the sample vacuum chamber. So typically our workflow involves doing a quick fluorescent mosaic of a grid to pick out cells of interest before going to soft X-ray tomography. And we can do things like track viral structures as a function of time, different time points post-infection, and at different time points post-treatment with antiviral drugs. And we can do some nice 3D volume segmentation. So just as a very quick overview of what it can do, just to capture our workflow in a little more detail, as I said, we can do a fluorescent mosaic. So here we have some yeast cells on a standard TEM grid. And then we can do the soft X-ray mosaic. So the third image to the right there is an 8x8 mosaic of soft X-ray mosaic taken with just 2D projections. So in a few seconds, we can grab a 2D projection, map the grid, make sure the cell still looks good, because you can't always tell from fluorescence. Fluorescence can make a cell look really nice, but when you look at it under soft X-rays, the cell can look very different. And then we can do the reconstruction, as I've shown here on the right-hand side. So we can flip through the volume of the cell. Essentially, similar idea with some Chlamydomonas algae we recently imaged for the Paul Scherrer Institute. We're picking out some nice structures there, such as the contractile vacuoles and the chloroplast and the pyrenoid, and of course the flagella going up really nicely. So how can we combine this with EM in a meaningful way, and what can it do for these workflows? We recently won a Marie Curie Doctoral Network Award, an EU-funded project, and we're working with 12 partners to develop sample transfer and data registration techniques. And basically, if you look at this picture from Rigord on the right-hand side here from his Nature Methods paper, where he depicts the cryo-ET workflow. We're seeking to bring soft X-ray in at this step here, along with fluorescence microscopy, and then you can go from there to on-grid thinning and cryo-ET, or you can start with imaging lift-out slabs, for example, which I'll show you in a second. So yeah, so you can do fluorescent soft X-ray, you can go to cryo-ET, vEM, or you can do free substitution and go to conventional TEM as well. So there's many different variations on this theme. And what do you get? You get an unbiased cell view, you get 3D volume context, and as well as reliable ROI targeting, where fluorescent targeting maybe isn't always available or super reliable. So we started doing some lift out of high pressure frozen slabs and doing soft X-ray tomography on these. So we started with Bacillus thuringiensis, if I'm pronouncing that right. It's a rod-shaped bacteria which consumes oxygen to produce these toxin crystals. So this was done with Michelle and Maud and the team here at RFI, which is a fantastic first step into this, validating that this workflow. So the bacteria slabs were a really nice starting point. So these were lifted out using the Thermo Fisher Hydra plasma fib and stuck onto rectangular grids. I won't go into the details of how the guys did it. Maybe they can explain this at another time. But I'll focus on what we could see once the samples came to Dublin. So these were shipped to Dublin in a dry shipper. Of course, the idea is you will not continuously ship samples to Dublin. The idea is we start shipping microscopes in the other direction. But for now, we're receiving samples in order to validate these workflows. And you can see we're picking out 3 slabs here in the integrated fluorescent microscope. We can see where they are. We can dial up their coordinates and then go to soft X-ray mode. And here we see 2 of the slabs, just in basic low mag X-ray projection mode. You can actually see the extent of the slab. They're about 30 by 30 microns, I think. And the guys, I think, yeah, they supply 2 different thicknesses, one 3 microns and one 6 microns, just to look at the effects of these. Yeah, so we can basically do the tilt series, run the tilt series, reconstruct the 3D volume of the slab, and then slice through it. And we can start to pick up the bacteria inside and the various structures. So these are quite a simple organism, of course, but they're a very good starting point for demonstration of the workflow. You can get really high contrast. You can see there's some ice picked up inevitably during the transfers. Yeah, just a zoomed in view, we can see that the crystals and in the spores. And the guys here, Maud, did a really nice segmentation of the bacteria where you can see the cell wall and some other red structure, which we think may be DNA. We're not sure, but time will tell. We extended this workflow then to worms, to worm larvae. So this is work done with the Plitzko group, with Christoph Kaiser and Oda. They did some fantastic work lifting out some slabs of the L1 larva. So you may remember, I'm sure you're familiar with Fitzko's seminal paper on serial lift-outs, imaging of the C. elegans worm, where he lifted out various slabs and then did fib thinning and then did cryo-ET and attempted to reconstruct the volume of the worm. Of course, doing it in this way necessitates actually fib milling away most of the volume. So you get a really nice data, but a lot of the volume is missing. So in this study, we're seeking to sort of recover or retain the volume of the worm, albeit at more moderate resolutions, and then just use your fib and do cryo-EM on selected slabs of interest. So we got some slabs from the green lines here are sort of towards the anterior. We still need to really figure out exactly where in the worm these are coming from, but essentially we have anterior leaning sections, and also the shaded region. We received some slabs sort of from the mid body towards the posterior as well, and these came from a waffle prepared grid, and on a 50 square copper grids, and these were lifted out using the Thermo Fisher Acrylis 2, and they extracted 5 micron thick slabs. And then going to our system, then this is the view from our fluorescent microscope inside the X-ray chamber. So we have some of the anterior, basically the 6 anterior slabs here on the left and the 5 mid-body slabs going beside it. We also got posterior slabs, but we didn't get a chance to look at these. This data is really just came out in the last couple of days. We had a look at these last week. So this is very, very fresh data. These are some stitched views of the 2D projection images from the soft X-ray microscope. So we can take a quick look at the slabs in soft X-ray projection, and then we can reconstruct or run the TIL series and reconstruct the volume of the slab. So that's one of the mid-body HAMO. So we went from 53 to minus 53 to minus 50 in 1 degree steps. The asymmetry in the angular extent is due to the fact that the slabs were slightly tilted on the grids by 10 or 15 degrees. And here's the anterior HAMO. So you can see some really nice structure within the worm. And I have some annotated images. So the idea then is, as I said, is to go downstream fib milling and cryo-ET on selected slabs, and just validate the integrity of the slabs following soft X-ray, validate the ice quality and the structural integrity in general. So excuse the busy annotations. This came out just last night. I got this from Christoph, so he worked late in the night to get me something. So well done to Christoph for getting this on time. It is a little bit busy, but the whole idea here is just to say that we can see lots of structure basically inside the slab. And this is the other one where you can see the cuticle nuclei. The large area in the middle is the intestinal lumen, etc. So that's that. We also have a related project. I'm going way too slow. Okay, a related project ongoing with the University of Nottingham with Chris Parmenter, and in this sense, he's using the Zeiss Crossbeam 550, so it's a slightly different approach. And instead of putting the slabs onto flat rectangular grids, we're putting them onto like a modified OmniPen, essentially. So we modified the OmniPen to be compatible with our microscope, and we have developed a puck. And the puck here can hold 4 of these OmniPens. The slabs sit on top of the pen, and we can stick them in a shipper or bring them to the lab next door or whatever. To do the soft X-ray imaging. We have some FibSim slice and view with roll and flex. So here's a bunch of Euglena algae on a grid, fluorescent mode, soft X-ray mode, and some of the soft X-ray images. These are complex organelles. So roll and fleck is interested in understanding or knowing whether or not really, or whether the structures of interest are actually there and whether it's actually worthwhile spending a few days fib milling through a whole Euglena algae. So he's using soft X-ray to give him that whole cell context before going to the FibSim. So here we have the same cell imaged in the soft X-ray microscope in Dublin, shipped back to London, and we have a FibSim movie here using the Joel FibSim. And he also did some cryo-ET on another cell This is the Golgi complex. So this particular cell was not imaged in our X-ray microscope, but it had been in our microscope for a period of time. So it still looks nice and intact. We have a project ongoing to do on-grid thinning with TESCAN and Anna Sartori and our team at Pasteur Institute and Dominic Pincus, Academy of Sciences. We've done one pass, which involved many sample transfers. We picked up some ice. We're doing a refined version of that again. And we can also go from cryo-SXT to and cryo-FM to room temperature TEM via free substitution, resin embedding, and sectioning. And we've done some work there with UCD Dublin. So on the left is a digital slice from the reconstruction, soft X-ray reconstruction, and on the right is a physical slice from the same cell. And we're just picking out some structures common to both. So that's all I have to say, except thanks to all of our many fantastic collaborators. I think this is really exciting. I think this could be really revolutionary in terms of, bringing soft X-ray microscopy to the lab. And I think the possibilities are endless in terms of what we can do. So, thank you very much.
Okay. Last chances for questions for today.
Thank you very much. Really interesting talk. I was wondering, given you had some of these mounted in grids, why you have very little beam hardening and shadow artifacts? Is it really monochromated source, or? In
the slabs or the grids?
Any that you had mounted in grids, so you had that copper, knocking about in the beam path, or was it not in the beam path?
I'm not sure what you're referring to.
Okay.
So yeah, so basically the beam itself is quasi-monochromatic. It's not fully monochromatic, but that's not leading to the effects you're seeing there.
So yeah, monochromatic is better. So that's why you don't have too much beam hardening.
Yeah. It's certainly spectrally it's broader than a synchrotron system, for example.
Sure.
But maybe what you're looking at is limited tilt artifacts as well. Not sure, but you get in cryo-ET anyway.
Yeah. Okay.
And actually, on that note, one of the things we want to do with Chris and with the guys here at RFI is to actually mill out a pillar instead of a planar slab, because our sample stage can rotate through plus or minus 90 degrees. So we can do full rotation tomography of fully hydrated tissue slabs. But I'm happy to chat to you afterwards in terms of specifically what you're referring to.
Really interesting. Thank you.
Thank you.
Hello. Do you have an example where your sample remains vitreous after imaging soft X-rays? And if so, how are you determining if it's diffraction afterwards, or is it just acquisition reflections? Or
Yeah, I think you can probably pick that up with in the electron microscopy data, if there's some devitrification there. And that's one of the things we're actually working on is to validate that. So for example, in terms of the slabs that we got from Munich, they kept some back in their lab as a control essentially. And they're going to do further milling and cryo-EM on the control slabs versus the ones that had been in our microscope. So then we'll determine to what extent the vitrification state is unchanged.
Hi. Yeah, sorry. I have more a naive question, and maybe this question could be addressed to all the speakers of the session, but when we're referring to X-ray imaging, some of you are referring to soft X-ray, another to hard X-ray. And so I understood that the sample preparation is different, but there is a difference regarding the resolution that we can get with which are the other X-ray imaging, and which is most of the difference, and why choose one versus the other.
Well, with soft X-ray tomography, it's typically limited by the zone plate you use because we're using the short wavelength light. and also the spectral bandwidth of your illumination system. So we can get down to about 50 nanometers, 55 nanometer resolution. I can't remember what the micro CTs are getting to, 800-ish, yeah. It's about an order of magnitude in the difference. And with soft X-ray tomography, it's really suited towards imaging cryo-prepared, fully hydrated, because you get that native contrast in hydrated samples, which makes it nicely compatible with cryo-ET workflows. Whereas with the micro-CT, you're typically imaging resin-embedded samples, which makes it compatible with conventional TEM workflows. Is that fair to say?
Anyone else? Okay.
I just have two technical questions. One is when you see the soft X-ray tomography, I see those black dots on the top whenever you do the tube series. And is this the labeling or is this some weight or whatever? And the second question is how fast you can get the data set.
So data sets typically take between 30 minutes and 2 hours, depending on the cell or the sample you're imaging. So it depends really on the amount of absorption, I guess, through the sample. So in terms of the yeast and the algae reconstructions I showed, it took about 30 minutes to acquire the till series. So that was plus or minus 60 degrees. And with some of the other samples, it might take 1 hour, 1.5 hours, whatever, up to 2 hours typically. So that's roughly the time span. I didn't quite get your first question, Alice, sorry. So they're probably lipid bodies, right? Yeah. So they're the most carbon-rich structures in the cell typically. So they absorb really strongly. So they really stand out.
It's good to give me a little exercise.
Not good for me.
So I wonder what are these limitations for the field of view? You mentioned it's 30 by 30 by 10. So the first question, 10, is it because you need to tilt the section and then sometimes it's limiting how much power you have on your source?
Well, it's a full field system, so our illumination spot is about 30 microns across. So that's really what gives us.
So you eliminate only like 30 micrometers by 30 area.
Yeah, roughly area. Yeah. And of course, you can change your magnification then, but yeah, that's really with the zone plates we're using. That's the field of view that works
so that the
really
data
nicely.
set that you collect is about like 512 by 512 pixels, or
about 2048 by 2048. Yeah.
Right.
So 2K by 2K detector, yeah.
Because I think you
So
said it's like if it's 30, so it's, okay, 30 by 30, you just focus the beam towards the spot, right? Then you want, you just kind of can't get the numbers right. So if I divide, let's say, 30 micrometers by 50 nanometers, so it's probably like about 500 pixels.
So it's magnification dependent essentially. So we can go to higher magnification, but we're not limited.